Okay, finally, as we talk about protein structure, we're going to talk about quaternary structure. Now, not all proteins have quaternary structure. Quaternary structure is a description of how different subunits of proteins interact with each other. Now, if the protein doesn't have any subunits, it's just one polypeptide, then it does not have quaternary structure. But a lot of proteins do. And so we're going to use some oxygen binding proteins, myoglobin and hemoglobin, as examples of two proteins, one that does not have quaternary structure and one that does. And we'll see the implications of the quaternary structure on the function of the molecule. So hemoglobin and myoglobin are very closely related to each other. We're going to call hemoglobin HB, myoglobin MB. They're evolutionarily related to each other and they're functionally related to each other. They're both globular proteins and they both have the ability to bind and transport oxygen through the blood. Myoglobin delivers it to the muscles and stores it in the muscles, and hemoglobin is in your red blood cells and delivers it all over the place from the lungs to peripheral tissues. Now, myoglobin is just a monomer, a simple polypeptide. It does not have subunits. Hemoglobin is a tetramer, right? So it does have quaternary structure, and it's a heterotetramer, meaning that all the subunits in the molecule are not the same. There are two alpha polypeptides and two beta polypeptides, and together these four proteins come together to form a functional hemoglobin unit. Now, while these are globular proteins, like we mentioned earlier at the start of our discussion about protein structure, they're not just amino acid based, right? This is a histidine amino acid that is binding to a heme group, right? The heme group is this complicated organic ringed structure that has nitrogens on the interior of it. The heme group also has an iron atom in the center conjugated to these nitrogens of the porphyrin ring. This organic molecule is called the porphyrin ring. And so we can think of this iron as almost being having octahedral coordination geometry to it. The four nitrogens of the porphyrin ring are bound in a square planar arrangement. The histidine of the amino acid chain of the myoglobin or hemoglobin subunit is bound in the fifth position, straight down, and in the sixth position, straight up, could be an oxygen binding. If oxygen's bound, we call that oxygenated. If oxygen is not bound, it's deoxy, right? Oxy and deoxy. So let's look at these proteins and see how they differ from each other. Here's myoglobin, right? Let's pop this up. Here's the structure of myoglobin, and it's a monomer, right? There's one polypeptide chain, and here we can see the heme group in the middle of it, right? This particular model has the nitrogens of the porphyrin ring outlined in blue, right? Nitrogen's usually blue. Here's the big fat iron atom in the middle, right? And then you can see the rest of the amino acid chain of this protein is mostly alpha helices, right? I'm not sure what these are. This may, I don't know what it's got here. This may indicate the N and C terminus of the myoglobin. Okay, well that's myoglobin. Here's hemoglobin, right? Let's make this a little bigger so we can see it. And we can see it looks huge, right? And it's almost like it's four myoglobins assembled together, right? Each of the subunits of myoglobin are different colors. Here, yellow, pink, blue, and green. And you can see that each of the subunits has a heme group with an iron atom in the middle. And so therefore, hemoglobin has the capability of binding to four oxygen molecules, while a molecule of myoglobin can only bind to one. Now, it's important, these subunits are distinct from each other, which means they're assembling together in this tetramer, but they are not binding to the other subunits through covalent bonds. They're just binding to each other through electrostatic interactions, right? Positives and negatives, or hydrophobic or hydrophilic interactions, um, but they're not covalently bound to each other. If they were covalently bound to each other, they would just be a single polypeptide, and we wouldn't consider them as separate subunits. Okay, so who cares about this, right? Hemoglobin does have quaternary structure, right? It functions as a tetramer, while myoglobin only functions as a monomer without any quaternary structure. Well, there's some functional consequences to the difference in the structure of these two molecules, and you can see them in the way they bind to oxygen. Here we've got an oxygen binding curve. On the y-axis over here, we're showing the percentage of oxygen that's bound, and over on the x-axis is the partial pressure of oxygen, right? So at low oxygen pressures, hardly any protein is bound to oxygen. At high partial pressures of oxygen, lots of high concentration of oxygen, most of your protein is bound to it. Okay. So let's look at the difference between myoglobin and hemoglobin in this chart. Myoglobin is pretty simple, right? At low partial pressures of oxygen, myoglobin is in the deoxygenated form. Now, as soon as you increase the partial pressure of oxygen, boom, most of your myoglobin is bound to oxygen. It's sort of pretty much a simple on-off scenario. Now, hemoglobin is a little different, right? Hemoglobin has what we call a sigmoidal curve. It looks kind of like an S curve. It's a little difficult to get that first oxygen bound to a subunit of hemoglobin, but then once one subunit of hemoglobin binds, the other subunits tend to bind really quickly, right? And that leads to this really quick increase in hemoglobin bound. It almost looks like a PCR curve or a bacterial growth curve. And the reason this happens is because these subunits in hemoglobin don't act independently of each other. They cooperate with each other to bind oxygen. So, totally deoxygenated hemoglobin, it's really kind of hard to get that first oxygen to bind. But once that first oxygen binds to one of the subunits of hemoglobin, the other subunits can bind to oxygen more readily. It's almost like a type of peer pressure, right? No one's bound to oxygen. I'm not going to do it. I'm not going to do it. I'm not going to do it. And eventually the concentration of oxygen reaches high enough that one of these subunits binds. Now, once one subunit binds, the other three subunits are much more likely to bind to oxygen. And once three subunits have bound to oxygen, that last subunit is under tremendous peer pressure and says, okay, sure, I'll do it, right? So these subunits are cooperating together. And it's sort of easy to describe as sort of a peer pressure. But how does that work really on a molecular level? Well, it turns out when hemoglobin, a subunit of hemoglobin, is unbound to oxygen in the deoxygenated form, it is in a particular shape or conformation called the T state or tense state. Now, when that subunit binds to oxygen and is the oxygenated form, it actually changes shape. It changes its conformation and becomes relaxed, the R state. Now, when a subunit goes from the deoxy to oxygenated form, or the T to the R state, that change in conformation, that change in shape, actually causes other subunits to change their conformation from the T state to the R state also, making them much more likely to bind to oxygen. Now, how does that occur? Well, here's a sort of a figure which shows what happens in this shape change from the T to the R state. I'll show you some more images of this in the classroom. But let's right now focus on this heme group. So in the blue here, we're showing the deoxygenated or tense form. In the tense form, the big iron atom is sort of shifted away from the plane of the porphyrin ring. You can see it here poking up above it. Now, when that iron binds to oxygen, we move from the tense form to the relaxed form, this pink structure here. And really that iron atom snaps right into the plane of that porphyrin ring as it binds to oxygen. And as that iron moves, it tugs at the histidine, which is part of the protein of that hemoglobin subunit. As it pulls on that histidine, it pulls on an alpha helix, and that changes the overall shape of that subunit of hemoglobin. As it moves from the deoxygenated in blue to the oxygenated in pink form, it changes its shape. And when it changes its shape, it actually changes those electrostatic, hydrophilic, hydrophobic interactions that bind these subunits together, right? Here's the way that the beta 2 and alpha 1 subunit are interacting together. You don't have to know the details of this, but this is how they interact in the tense state. Now, once oxygenation occurs on the beta subunit, it moves it. It changes its conformation. And what that does is it changes the way it interacts with that alpha subunit. Now, what that does to the alpha subunit is it causes it to adopt that relaxed state, even though it's not bound to oxygen. And when it's in that relaxed state, it's much more likely to bind to oxygen than it was when it was in the tense state. So this is sort of a molecular description of how that peer pressure occurs. We have one subunit giving in to higher partial pressures of oxygen, binding to oxygen, going from the tense to relaxed state, and that sort of bumps against its neighboring subunit, causing it to adopt the relaxed state and causes it to bind to oxygen more readily. And that is the cooperativity that we see due to the quaternary structure of hemoglobin.